If you have a developmental condition, then it will be with you for the rest of your life, and it will affect you and your family and society for many, many years. Developmental disorders are very common. 1 in 100 of us develops schizophrenia, 1 in 68 autism, 1 in 30 attention deficit hyperactivity disorder, and 1 in 10 of us have dyslexia. The cells which are generated in the embryonic brain, they have an address and they have a destination, and they already have a fate. Now, if these cells do not arrive to the right location, then you can have all sorts of developmental abnormalities. So it's essential to understand how these cells are generated and how they migrate. One of my major research interests is a transient cell group in the so-called subcortical plate. And these cells form the basis of the cerebral cortex, and the cerebral cortex will develop above these transient cells. But without these cells, you could not set up the cerebral cortex. And after they finish their function, these scaffold cells, they get dismantled and they disappear with programmed cell death. So there's no point studying the adult. You don't see these cells in the adult. You have to study the developing brain to understand what can go wrong. And most of them will disappear, about 80–90% of them will disappear by the end of the first postnatal year. However, if you have a difficult birth, hypoxic ischemia, these cells are very sensitive for hypoxia. They will disappear and you cannot finish the building work. And your brain will be very different. For instance, cerebral palsy can develop, and these conditions can be with that person for the rest of their lives. So it is important to understand these transient cells, these transient connections, and the principles of brain development if we want to intervene with these conditions which can have an impact on the rest of our life. We are beginning to understand the long-term consequences of the developmental abnormalities. So we are looking at these conditions lifelong. My vision on developmental neuroscience is to link these very early steps to later conditions. And I think we have a real chance to do that through the UK Biobank or the long-term, lifelong follow-up of conditions, and backing all this up with very solid genetic data. There was a revolution now in studying human brains with the amazing imaging techniques which you can use even in utero, using MRI or ultrasound. And you can even detect connections in the developing brain using all sorts of analytical methods with MRI. The Institute of Child Health and also Newcastle University Medical Research Council and Wellcome Trust established biobanks, and they really help us to get an insight into the real development of the human brain, rather than looking at small organoids which were generated from induced pluripotent stem cells from your skin. So it's very important to do both, not just looking at these artificial systems, but also the real brain. When the young generation is reading research papers, they usually just read, the end product. They don't see all the sweat and dead-end streets we had to go through. So the first advice I would give is to be prepared to fail, and then you have to correct the research direction. So take the initiatives, but don't be afraid to fail. And the second is to be confident in your research questions. So if you have a fundamental, very important research question, then don't get distracted with the technical details. Go for the question and try to answer that. The third is to select good mentors. I was very fortunate that I had exceptional mentors both in Hungary and here in Oxford, and also in Lausanne in Switzerland where I worked. And this helped me to get through the difficulties. Our research is guided by research questions and not by techniques. So we have to be able to juggle very different techniques like bioinformatics, genetics, anatomy, histology, electrophysiology, and also high-level imaging You have to collaborate. You cannot accumulate all these skills in a smaller or medium-sized laboratory. You have to collaborate, and that's the beauty of Oxford. You have a huge variety of different experts, and they are very happy to collaborate, and there are excellent students and excellent colleagues, and Oxford is a superb environment to work in. So the great challenge is to be able to combine very different technologies in our research. So if I had a blank check, I would just distribute it among these groups and foster more collaboration.